This is a diploid cell in interphase. It's diploid because there's two copies of each chromosome, one from the mom, one from the dad. In prophase, the chromosomes will duplicate and form homologs.
Say something. You can say something. All right.
Next, the chromosomes will cross over.
Should we mention something?
Do you want to do
Sister chromatids.
Yeah, these are the sister chromatids in the homologs. They're both sides. All right.
Next, the homologs will line up at the metaphase plate, which is like halfway through the cell.
Metaphase
Well,
1 and meiosis 1.
The nuclear membrane is
It's gone.
breaking.
It's
Yeah,
gone.
it's gone.
It's breaking
So
down,
we
and
erased
then
it.
the centrosomes will, with the microtubules will separate the homologs, and in this stage there's the law of independent assortment, which means
In
like,
metaphase, there's the law of independent assortment. They're still together.
but they have to come.
This
Sure.
is metaphase. So the law of independent assortment is basically in what order they're lined up, which is a source of genetic variation, as well as the crossing over is also a source of genetic variation. And then we go into anaphase 1.
In anaphase 1, the homologous chromosomes separate, but the sister chromatids remain together, and so they separate to their different parts of the cell. This is the law of segregation, so that the two chromosomes separate. In telophase 1 and cytokinesis, the nuclear membrane pinches off, so it's more like it's becoming to look like two different cells, and then the nuclear membrane starts forming.
Mm-hmm. And the law of segregation happens during anaphase 1, which is them pulling apart and is another source of genetic variation. So these are now two separate cells.
Okay, so in prophase 2, there are two different haploid cells. Each here, you can kind of see them. They're separate, completely separate now. Both still have chromosomes with sister chromatids attached, but they're not genetically identical because the homologs have separated into the different cells, and the chromosomes won't duplicate again after this. In metaphase 2, the chromosomes line up at the metaphase plate for each cell, which is about halfway in the middle. And the chromatids are not genetically identical because of crossing over, as you can see here. So these won't be genetically identical to those.
All right, then we move into anaphase 2, where these separate, forming four haploid unique germ cells. There will be 4 separate, and you can see that they're each unique. So depending on which egg cell and which sperm cell come together, it will make a completely unique zygote. So it pinches off in telophase and cytokinesis, making the 4 cells. And that is meiosis. Is there anything else?
These are haploid cells.
Haploid cells. It's because they only have one copy of the mother's and chromosome and one copy of the father's chromosome. They're all unique.